I am Sohail Mujiri. I'm doing my PhD in 3rd Institute of Physics, Göttingen University. My PhD is focused on multiplane 3D super resolution optical fluctuation imaging. It's called 3D SOFI. SOFI is a simple post-processing technique to resolve images beyond the diffraction limit of visible light. It is simple since we just need to take a short movie from our biological sample labeled by blinking emitters like fluorescent proteins, some photo-switchable fluorescent proteins. The setup is based on a conventional Light-Sheet Microscope. In comparison to other existing super-resolution techniques, SOFI can be employed to image a wide range of biomedical samples. In principle, SOFI can deal with different blinking timescales of fluorescent labels and high-level densities. In order to avoid phototoxicity, photobleaching, and record a fast dynamic process inside the cell, we need a fast imaging technique. Instead of scanning through the sample, we are implementing a multiplane MIMO splitter in our conventional wide field setup to be able to image all the focal planes, in our case 8 focal planes, in the same time. Our main goal is 3D super resolution live cell imaging. Now I'm going to show you the 8 distinct planes through the sample. It's interesting to know that with correlating between these planes, we could enhance the axial resolution even more.
My name is Adrián Escobat and I'm doing my PhD at the University of St. Andrews. In our research group, we develop new microscopes designed to visualize the life processes that take place in our bodies. We work in close collaboration with researchers of different fields like biologists or doctors with the common goal of understanding the main causes of devastating diseases like Alzheimer or cancer. In order to do that, we image model animals like mice or zebrafish. My research project is mainly focused on light-sheet fluorescence microscopy. In this technique, a single plane of the sample is illuminated with a very thin sheet of light. By moving the sample or the light sheet axially, we can obtain fast three-dimensional images with high depth resolution and low photo damage, because we are only illuminating the plane of interest.
And now we're in the lab. This is what the front of a Light-Sheet Microscope looks like. This is the illumination path, the detection path with the camera, and the two orthogonal objectives. The sample is placed in a chamber sitting on top of the stage. In the experiment that I'm currently doing, I'm imaging a living zebrafish, and on the screen you can see its beating heart.
I am Mariano González Pisfil, a BeOptical early stars researcher at Humboldt University of Berlin and Picquecont. My PhD project consists in developing a novel approach combining super-resolution microscopy and time-resolution microscopy to study molecular dynamics in living cells. With the advent of fluorescent proteins, the field of photonic microscopy has grown exponentially in the last decades, improving our understandings in cell structures and mechanics. Among them, cell membranes are intricate structures with many roles. For example, they protect the cells from external aggressions and participate in cell-to-cell communications. To be able to fulfill its different roles, they are made of a complex arrangement of lipids and proteins unique to each cell type. Even if photonic microscopy has greatly enhanced our knowledge about them, due to the wave nature of the light, it was not possible to track or image objects smaller than 250 nanometers. This changed with the appearance of super-resolution microscopy techniques. Among them, with STED or stimulated emission depletion microscopy, we are able to shrink our observation volume up to 30 nanometers, dramatically increasing our resolution level. Although multicolor STED microscopy is still challenging. So the project aims to combine STED with time-resolution microscopy in which picosecond laser schemes and cutting-edge detectors are used, not only to be able to image multicolor stat image, but also to study the dynamics of the samples with a high spatial resolution. The Microtime 200, a state-of-the-art stat microscope, will be used for this project. Multicolor stat of cell membrane dynamics will lead us to a better understanding of how plasma membranes are structured and respond to stimuli.